Well, I think this session is going to explore the really broad questions of drug targeting and drug discovery. There'll be my talk, which will consider some of the systems approaches to antibody use of drugs and things like that. And there'll be several other talks that will focus on either specific examples of antibodies that are drugs and or how you produce antibodies that can be drugs. I think that the big opportunities for the industry are to make antibodies more stable, less cross-reactive, and to be able to produce them in a more efficient fashion than we can now. And of course, for the longer future, there is a possibility there'll be other protein capture reagents that can be used as drugs that may be even more effective than antibodies. Well, I see industry heading toward the production of protein capture reagents that are more stable, have less cross-reactivity, have higher sensitivity and specificity. Whether this is going to be the enhanced use of antibodies or whether it's the creation of new reagents, peptide protein capture agents, aptamers, or the like, I think is very uncertain now, but I think it'll be a very exciting time. I think the biggest challenges are, frankly, probably not going to be discussed very well in this conference. I think the biggest challenge is, one, can we diagnose patients with disease accurately? Two, if we get a disease, can we stratify it into its different subtypes so we can create a proper impedance match for drugs that are really effective? And three, what are the targets that we should direct our antibody-like reagents at? And finally, four, how do we really effectively make the drugs, make the antibodies that are as stable as possible and efficient, minimize cross-reactivity, and the other issues that plague any therapeutic reagent? I think the opportunities are, with the advent of systems biology, to use that systems biology to do exactly what I said: the diagnosis of disease, the stratification of disease, and most important of all, the appropriate choice of targets. So I think there are enormous opportunities for the future. And then there is, of course, the fourth challenge of how do you make effective drugs?